Good morning, student. Today we are going to know about Basidiomycotina, that is Puccinia graminis. So first of all, Puccinia graminis is nothing but an fungal pathogen which depends their complete life cycle in two different plants, that is a sugarcane and barberry plant. This Puccinia is included in class Basidiomycotina. So these are the two different stages, and this is a cross section through barberry plant, and this one is the cross section through sugarcane plant, and this was the complete life cycle of Puccinia graminis, which contributes near about 5 different types of spores. So in case of a distribution, Puccinia counted as 700 species which are found all over the world, while 150 species which are reported from India. Many of them are host specific. What is meant by host specific? To complete their life cycle, they need specific hosts, as like sugarcane, barberry plant, or whatever it may be, another plant. Due to its pathogenic attack, a crop plant going through enormous losses are caused by the fungal genus, that is Puccinia, while this macrocyclic rust disease consists of different 5 kinds of spores, that is Uredospores, teliospores, basidiospores, pycniospores, aeciospores. So it is a heteroecious, means hetero means different, different types of 5 different types of spores they can develop, means the spore stages are produced on 2 different hosts, that is wheat and barberry. So first of all, this is the diagrammatic representation of a life cycle. When Puccinia graminis infect wheat plant at that time, it can create many more different type of symptoms, and within that symptoms there are most important, that is a development of a noticeable red color patches on leaves, leaf sheaths and stems. So this is the cross section through the leaf, and these red filaments are nothing but an fungal hyphae, and here is the development of spores, so that spores are going to be come outside of the leaf epidermis. At that time, that fungal hyphae can create such type of uridium. That uridium possesses few to many number of Uredospores. So these are the Uridinospores. What happened, that Uridinospores are transferred with the wind and again placed on another plant body, and at that time they germinate into a teliospores. These are the brown color teliospores, and this is the brown color filamentous structure is nothing but a hyphae, and with the near that hyphal filament there is a circular dark spot are nothing but an hostoria, and this is nothing but an teliospores which are liberate out through the epidermal wall. These teliospores on germination they produce promycelium. So these are the single filament which can going to be developed into a mycelium, and these basidiospores again, that teliospores germinate into a basidiospore. That basidiospores again infect the barberry plant, and within that barberry plant there is a development of 2 different type of spores, that is pycnium can develop into a pycniospores, aecium that can be developed into a aeciospores. So the pycnium is nothing but this triangular outgrowth which shows many of pycniospores, and here is the aecium. That pycnium is mature into a aecium and they develop a aeciospores. Again that aeciospores can germinate on a sugarcane plant and develop a uridinium, and uridinium consists of Uredospores. So in this way Puccinia gramini tritici can complete its own life cycle in this way. So classification is nothing but a hierarchical arrangement of a different taxa. So first of all, division. Puccinia gramini can included in a Eumycota, then subdivision, that is a Basidiomycotina class, that subdivision Basidiomycotina classified into a teleomycetes, and again they are ordered into a Uridinals, and finally it can be included in a family Pucciniaceae, and the genus is nothing but an Puccinia. Then species is a Graminis. So in this video we will discuss about the life cycle of Puccinia graminis with the help of suitable diagram and Dikaryophase in the life cycle of Puccinia. So this phase in the life cycle of Puccinia is confined to the primary host, which is a weed. It consists of dikaryotic mycelium and two spore stage, that is uridinal and teleto. So here is the many more different types of mycelium, which is a dikaryotic mycelium. It is internal and thus invisible until it ready to reproduce. Means what? This mycelium is present inside the cells of a leaf. That leaf are naturally of a sugarcane, and when that mycelium can be developed into a reproductive bodies as like a spore body, at that time they are visible. The binucleate secondary or dikaryotic mycelium, which is filamentous and well developed and branch, means that mycelium. So here is the diagram which can shows mycelium. This is the red color long elongated filaments are nothing but an mycelium, and here is the small outgrowth, circular outgrowth, that is a hostoria. This one and this one are the mycelium, which is a dikaryotic binucleated, while the hostorial apparatus, which are reported by Allen in 1923. He reported that the tips of certain hyphae of intercellular dikaryotic mycelium come in contact with the cell wall of the host cell and become separated from remainder of the mycelium by septa. So over here there is a presence of septed mycelium with the hostorial apparatus. The resultant small binucleate tip cell swell somewhat and function as a hostorial mother cell. That hostorial mother cell at the point of contact, each hostorial mother cell develop a bulk-like thickening which is closely appressed to the host cell wall. So here are the many more morphological as well as developmental characteristics of the hostorial apparatus. Then hyphal wall of the plasma membrane of the hostorial mother cell are continuous along the entire length of the penetration tube and hostorial body constituting the hostorial wall and hostorial membrane, that is below each other. Then collar-like structure attached to the host cell wall and made of the material is produced by the host cell in response to the presence of penetration tube. It surrounds the penetration tube at its proximal end. This hostorial body is again binucleate on the outer surface of the hyphal wall. In the body region only is formed an electron dense sheath matrix consisting of a granular amorphous material. This sheath matrix is also designated as the extrahostorial sheath. This fungus obtains nutrition from the host, which is infected wheat plant exhibit stunted growth. Means when this pathogen infect wheat plant, at that time the growth of the wheat plant going to be stunted and they could not be grow into a major heights. When we are taking a transverse section through septa between the cell have each a central pore without diplopore parenthesum complex. The clamp connection however have not been reported on the dikaryotic mycelium. This mycelium is generally localized. It grows and ramifies near the point of infection, extending only short distance into the host tissue. The fully developed dikaryotic mycelium enters upon the reproductive phase. So this is all about hostorial apparatus, mycelium. Then we are going to know about reproduction in a Puccinia graminis. So first of all, here is the diagram which can shows uridinal stage of Puccinia graminis. So A is nothing but an Uridosori on wheat leaf. So this is the wheat leaf which can shows Uredospores, that is nothing but an red color markings with these circular patches. Then B is a section through the Uridosori, means Uredospores. This is this part which is a bulky in nature. When we are taking a cut through this transverse side, at that time they can show this type of structure. First of all, they possess a host epidermis. Below that host epidermis, they have a mycelium. This is nothing but a hyphae, and these are the stalks of Uredospores, which are again binucleated. So this is the single spore which are grouped into these Uredospores. So this is a circular body, that is a spore, and this is the stalk of the single Uredospores. So what happened at the time of reproduction? It is a dikaryotic mycelium which produced by the sporulation. Sporulation is nothing but a spore formation. The spores produced are of two kinds, that is a Uredospores and Teloto Spores. They are produced near the surface of the host tissue. When mature, they break into a slits or pustule called as a sori. The mature spores are thus seen externally in this cross section. Then Uridinal stage. Prior to the Uredospore formation, the hyphae of the dikaryotic mycelium begin to aggregate near the surface of the infected organs such as leaf stem, leaf sheath, glooms of the hyphal mass which surrounds isolated host cells. So this is the B section or B diagram, which shows mass of surround isolated host cell. The mycelium hyphae then produce mass of cells subepidermally. These are called uridia, means this complete structure is called as uridia, and single spore is called as a Uredospores. From the uridia arise vertically growing cylinder stalk-like hyphae arranged in a close palisade-like layer. So this is long elongated palisade cells. The tip of each hyphal stalk or Uredospore mother cell wall swells, means at the top of the hyphal stalk there is a presence of Uredospore mother cell which is swelled to form a single binucleate oval Uredospores or Uridinospores. Uredospores are thus formed in a group. Each such group is called Uridosori or Uridinum. In this way they are going to be developed and attains their reproductive structure. Later on they are going to be germinate. So germination of Uredospore and infection of the host plant. So within this diagram here is the Puccinia gramineae which can shows a diagram, that is sorus with both Uredo and Teloto spores. So this single circular body is nothing but an Uredospore, while this two nucleated structure is nothing but an Teliospore. The diagram B is nothing but an germination of Uredospore, while C is the infection of wheat leaf by germ tube through a stoma. Stoma is nothing but an stomata. So here is the stomatal opening. This stomatal opening can be triggered with the germ tube, and that germination is start over there, and the hyphae or mycelium can grow up and take a entry into the palisade parenchyma. So this is the palisade parenchyma, and these are the cortical cell, and within that complete complex this is the mycelium, which is a septate binucleate mycelium of a Puccinia. So in this way they are germinate into a host plant body. Then this is the teleal stage. Late in the growing season, another kind of spores begin to develop from the uridia in the Uredosori. It is the Teloto spores. Teloto spores are first developed among the Uredospores in the same sori, and they are of dark brown to black in color. Generally, as the season progresses, more and more Teloto spores are produced, whereas the number of Uredospores is reduced. Means when the Teloto spores are developed at that time, they are developed more and more, while the Uredospores development going to be terminate, and that's why Uredospores growth is going to be reduced. Finally, the sori contain only Teloto spores, and these sori are called as Telotosori. So the cells of the Teloto spores exert pressure on the overlying epidermis, which is at first lifted but finally ruptured. Means first of all, that's epidermis having its own strength, but finally due to the enzymatic activity that epidermis rupture, and the Teloto spores are going to be attack or take entry into the epidermis. So in this way they are going to be germinate. So this is all about the germination or development of a Teloto spores, and After the completion of a telial stage, there is a starting of a basidial stage. The basidial stage is nothing but an favorable condition for the Teloto spore germination. So within a basidial stage, there is a germination of a Teloto spores, and at that time there is a production of a basidial stage in the life cycle of a Puccinia. The characteristic structure of this stage are the basidia and the basidiospores. So this is the hypobasidium and this is the epibasidium. Here is the basidiospores and this is the germination of Teloto spores and basidial stage. Basidium in a Puccinia is visible into three parts, that is a probasidium or hypobasidium. So this is a hypobasidium. Then epibasidium, this A and B is the epibasidium. The sterigmate which bears the basidiospores. Sterigmata which bears the basidiospores. Here is the sterigmata which can develop into a basidiospores. Each cell of the Teloto spore containing a diploid nucleus represent the probasidium or hypobasidium. At the time of germination, a short cylinder hyaline hypha of limited growth and promycelium or epibasidium grow out through the germ pore of the each cell, that is probasidium of the Teloto spores. So in this way Teloto spores are germinated into a basidial stage. So here is the many more consequences of a basidial stages or the development of a basidial stage into a basidiospores. Again, that basidiospores are taking a suitable alternate form and develop into a further new body. So here is the germination of basidiospores and haplophase of the life cycle of a Puccinia graminis. In this phase, the life cycle is confined to the alternate host, that is a barberry plant, that is a Barberry vulgaris. It starts with the basidiospores, means the basidiospores are transferred from a sugar cane plant with the help of wind and placed on or directly placed on or liberated on barberry plant, that is a Barberry vulgaris, and at that time haplophase consists of primary or haplomycelium and the two spore stages. These are the spermatogonial, also called as a pycnidial, and then another one, that is a aeciospores stages. So here is the diagram which can show a detail, that is Puccinia graminis, that is a spermagonal and aecidial stages. So A is nothing but a vertical section of a barberry leaf showing a spermatogonium, that is a pycnidium, on the upper surface. So here is the pycnidiospores or spermatia, which is having receptive hypha and a acial cup on the lower surface. So here is the development of acidial cup or circular cavity on the lower surface, while the B part is nothing but an showing mode of development of acidiospores. So here is the single aeciospores going to be germinate into a intercalary sterile cells, and this is the stalk. So this is the vertical arrangement of a acidiospores. First of all, basidiospores germinate into a haplomycelium and then they are attack on the alternate host, that is a Barbaris vulgaris. So here is the some consequences of the development of a pycnium and aeciospores. So here is the mature spermagonium. In this way, spermagonial stages are going to be complete, and that spermagonium arise with three kinds of hyphae, that is a spermatiophores, periphyses, flexus receptive hyphae. So here is the barberry plant which has a fungal infection of a Gramini, a Puccinia gramini. At that time they can arise, they can develop three different kinds of hyphae. First of all, that is a spermatiophores. So it is a numerous fine, long, elongated, uninucleated, short hyphae which are arise from the cell wall, and they project into the cavity of spermagonium and called as a spermatiospores. So then there is a second type, that is a periphyses. They are long, delicate, sterile hyphae which develop near the ostiole from the spermagonial wall. And here is the flexus receptive hypha which is adjacent to the ostiole and below or among the stiffer tapering periphyses which develop another kind of hyphae. They are slender, delicate, cylindrical with blunt free tips. These flexes are named the receptive or flexus hyphae because they are septate and may be simple or branch. Each septum has a central spore. The nature of spermagonia and spermatia has long been debatable point. It was in 1917 that Carnegie Buehler and his associate demonstrated that spermatia function as a male cells, while the receptive or flexus hyphae are supposed to be function as a trichogyny hyphae. They develop late and are spermatized by the spermatia of a opposite strain. So the spermatism is nothing but an opposite sex which is having a positive bar and negative bar. So these are the some details about spermatium, and after that here is the another stage, that is a acidial or acidial stage. In this stage, here is the infected area of the leaf of a barberry plant. So Puccinia graminis, a basidiospores directly germinated on a leaf of Barberry vulgaris and infect leaf of it with acidial cups on the lower surface. This is the acidial cup, and this is the enlarged part of the acidial cup. So here is the some detailed structure of a acidial cup. Then the development of aeciospores. At the time of aeciospore formation, binucleate aeciospore mother cell increase in length. The two nuclei in it divide conjugately and small daughter cells is then cut off at its terminal end. In this way they are going to be developed, and here is the some developmental details are reported over here. Then this is the complete diagrammatic representation of a Puccinia graminis. So this schematic representation having 5 different stages of spores which are complete their life cycle on 2 different host plant, that is sugarcane and Barberry vulgaris. So This is the diagrammatic representation which shows diplophase, haplophase, dikaryophase. This complete life cycle is on a barberry plant, while this complete life cycle is depend on the first pathogen, that is a sugarcane. Here is the germination process of aeciospores, then life cycle pattern of Puccinia, that is completely depends on 5 different spores, that is a spermatia, which is a unionucleate in spermagonia on barberry plant, then acidiospores, that is a binucleate on acidia on barberry plant, then Uredospores, which are binucleate dikaryotic mycelium on a wheat plant, Teloto Spores, binucleate dikaryotic mycelium on wheat plant, basidiospore, uninucleate on Epibacilla. In this way they can complete their life cycle. So many more different types of details are recorded over here. You can read it one by one, and this is another different type of a diagrammatic representation of a Puccinia life cycle. So this is all about a Puccinia, and here is the alternation of generation of Puccinia. Many more details are written over here. You may read out in one by one. Here is the again few important diagrams. This is the complete life cycle of a Puccinia. This is the barberry plant and this is the sugarcane plant. So thank you for watching my YouTube video lecture.